Hello, I'm Seshnag and this video is my subject-specific task for biology. In this video, I'll be covering the topic of gel electrophoresis while walking you through a virtual lab provided on the same. Nearing the end of the video, I'll also be discussing northern, southern, and western blots. Now, before I continue, have you ever wondered how detectives use DNA to track down criminals and suspects? I mean, there are loads of different methods that forensic scientists use to identify a criminal, but one of the most basic ones is gel electrophoresis. Now, without any further ado, I'll get on with it. Gel electrophoresis is a technique that utilizes the charged nature of particles to separate molecules like DNA, RNA, and proteins according to their size. The use of electrophoresis began with the work of Arne Tiselius in 1931, while new separation processes and chemical analysis techniques based on electrophoresis still continue to be developed in the 21st century. Tiselius invented the Tiselius apparatus, which was used for moving boundary electrophoresis. This is a type of electrophoresis that I will not be covering in this video, but it sure is a topic for further research as it is very interesting. Now, history aside, in gel electrophoresis, an electrical current is applied across the gel so that one end of the gel has a positive charge while the other end has a negative charge. The gel consists of a permeable matrix, which is a bit like a sieve, as you can see, that negatively or positively charged molecules can travel through. When the molecules pass through the gel, the smaller fragments migrate more quickly and travel further than the larger fragments that migrate more slowly and hence travel a shorter distance. As a result, the molecules are separated by size. Now, how does all of this work? Let's just look at DNA for now. DNA molecules have a negative charge because of the phosphate groups present in their sugar phosphate backbone. So, they're able to move through the matrix towards the positive pole, as you can see in the virtual lab. Once these molecules are separated, scientists use dyes, fluorescent tags, or radioactive labels, which enable them to see the DNA on the gel. The DNA is observed as well-defined lines, which are called bands, once they've separated. Now, when running a gel, a scientist uses a similar DNA sample to the one being tested. This sample has known lengths of DNA, and it's used as a standard to determine the fragment sizes of the DNA being tested. It's also known as a DNA ladder. Now, don't be fooled. In this virtual lab, there are only 6 pieces of apparatus used for conducting the experiment. In real life, there are a few more chemicals and apparatus used to conduct this experiment. Let's talk a bit about the chemicals that are normally used in agarose gel electrophoresis. First and foremost, we have agarose powder. Agarose is a polysaccharide extracted from seaweed. The long chain of polymer known as agarobiosis creates this agarose sugar. For this application in gel electrophoresis, a chemical known as agaropectin is removed from the agar to form agarose. When the agarose is dissolved and heated in a buffer, it begins to undergo hydrogen bonding, and as it sets in the gel mold, it begins to form a three-dimensional matrix of pores. Now, to control the size of these pores that the gel forms, scientists use various different concentrations of agarose powder to make the gel. As the concentration of the powder increases, the size of the pores within the gel decreases. Before the gel sets, a gel comb is inserted at one end of the gel mold. This is to create wells in which the DNA will be inserted into during the experiment. Once the gel sets, the gel comb is removed and the electrophoresis chamber is prepared by pouring in a buffer solution. This buffer acts as an electrolyte, allowing the movement of electricity through the gel, and it also prevents the gel from drying out during the experiment. Before the gel is placed within the electrophoresis chamber, the DNA has to be prepared with a loading dye as well as a buffer. The buffer facilitates the liquid medium for the migration of the DNA into the gel, and it also maintains a constant pH during the run. Normally, a buffer used in DNA gel electrophoresis is TAE. TAE is a mixture of tris base, acetic acid, and EDTA. Or a buffer known as TBE, which uses boric acid instead of acetic acid, can be used as well. Now, the next chemical used to prepare the DNA is the DNA gel loading dye. This dye is what adds color to the DNA, as it normally contains bromophenol blue and glycerol. The glycerol present in the dye gives density to the DNA, which allows the DNA to settle at the bottom of the well, and the bromophenol blue runs ahead of the DNA so that a lab technician can monitor the migration of the DNA through the gel. Once the DNA is prepared to be loaded, a micropipette is used to load the DNA into the wells of the gel. After the DNA being tested is loaded, a DNA size standard, the DNA ladder, is also loaded into the gel next to the DNA being tested. Once the gel has been loaded, the electrophoresis chamber is connected to a power supply, after which the gel is run from about 1 to 1.5 hours. This is the typical runtime of gel electrophoresis, depending on the gel concentration and voltage. A typical voltage which is used for electrophoresis is 80 to 150 volts, and the gel is run until the dye line is approximately 75 to 80% of the way down the gel. While the gel is being run, the DNA migrates towards the positive end of the gel. The short DNA strands move more quickly through the holes in the gel, whereas the long strands take a longer time. The shorter DNA ends up closer to the end of the gel, whereas the longer DNA ends up closer to the wells. Once the gel has been run, the gel is removed from the mold and placed into a DNA staining solution. This staining solution has a chemical called ethidium bromide. Ethidium bromide is a fluorescent dye used in tagging DNA. As DNA doesn't have its own color, ethidium bromide intercalates with the DNA bases and gives a fluorescent color to the bands formed on the gel when exposed to a UV light. Once the gel is removed from the ethidium bromide solution, it's patted down with a few tissues and placed on the UV light. Now we've near the end of the virtual lab. The last activity that is provided is a small activity where you can fill in your size estimates for the DNA sample. Note that the estimates that I filled in are wrong, as I kind of messed up during the recording of this video. But yeah, have fun doing the virtual lab yourself. Now that you've done gel electrophoresis with DNA, there's a little bit more that you ought to know. What we've just done is a simplified version of a southern blot. There are various different types of blots, but there are three main blotting techniques: the southern blot, the northern blot, and the western blot. These different blotting techniques are used to identify unique proteins and nucleic acid sequences. The southern, northern, and western blot protocols are similar, as they all require an electrophoretic separation of proteins and nucleic acid fragments in the gel. Usually, blotting techniques are selected based on the target molecule, which can be DNA, RNA, or protein. What you see here is a southern blot used for DNA. What we did in the virtual lab is a simplified version of the southern blot process. Now, southern blotting also has a few more steps after electrophoresis. The DNA on the gel is transferred to a nylon membrane, which is then incubated with a nucleic acid probe that has a sequence homologous to the target sequence and is labeled with a radioactive fluorescent dye, or an enzyme capable of generating a chemiluminescent signal. This nylon membrane is then visualized with an X-ray film or phosphoroimaging. And that's all there is to a southern blot protocol. Next, we'll talk about the northern blot when it comes up. I'll see you then. Now, let's talk a bit about northern blots. Northern blots are used to determine the identity, size, and abundance of a specific RNA sequence. These protocols normally begin with RNA isolation, and the separation techniques vary depending on RNA size. For example, large RNAs are separated by electrophoresis on a formaldehyde agarose gel or a glyoxyl agarose gel. These gels prevent normal base pairings and maintain the RNA in a denatured state. Once the gel has been run, the RNA is transferred from the gel to a nylon membrane, which is then incubated with a radioactively or non-isotopically labeled RNA or DNA. The unhybridized probe is then removed by washing with a buffer, and the radio-labeled probes are visualized with X-ray film. Last but not least, we have Western blots. This blotting technique is used to determine the identity, size, and abundance of specific proteins within a sample. The Western blot protocol begins with a sample lysate preparation from a tissue or cell culture, and the separation of the protein on a polyacrylamide gel via electrophoresis. The separated proteins are then transferred to a nitrocellulose or a polyvinylidene difluoride membrane, a PVDF membrane. It's a bit of a mouthful to say. The membrane, the membrane is then incubated with a blocking agent to prevent non-specific binding, followed by incubation with a primary antibody to bind the protein of interest. There are two detection methods used: direct and indirect. Direct detection relies on a labeled primary antibody, whereas indirect detection requires a primary antibody directed against the target protein and a secondary antibody directed against the immunoglobulin class or subclass of the primary antibody's species. Visualization of the final product include methods such as colorimetric assays or chemiluminescence. As we near the end of this video, thank you for sticking around with me while exploring gel electrophoresis and its various blotting methods, and stick around for the citations near the end of this video. Thank you and bye. My God, I've been sitting here for a bit over an hour.